obviously studies genetic variation, that is to say mutations that arise in DNA sequence, and we learning about the essentially properties and consequences of these mutations, and so that's what I'll be talking a bit about here today. So by way of introduction, many or some mutations that occur in genomes may have an effect on reproductive fitness. So that is to say individuals with a particular mutation may have more children than individuals who don't carry a particular mutation. And that's an example here of something that we call positive natural selection, where we might have a beneficial mutation arises in the genome, and if it becomes established, it goes on to reach high frequency in the population. Another kind of selection is what's called negative natural selection, and this is essentially the opposite process, where here the mutations are deleterious and reduce reproductive fitness, and as such, individuals who carry a particular deleterious mutation tend to have fewer children, and thus these deleterious mutations, at least in large populations, are reduced in frequency. And so the goal here is to try to understand a bit about natural selection from genetic variation data. So to look at this a bit more quantitatively and precisely, this is what I think is one of the most important distributions in all of biology. That is something called the distribution of fitness effects, or DFE, and I'll be talking a bit about this and how we can study it in my talk here today. So basically, the idea here is that we can imagine if we put a mutation into a genome, that particular mutation may have some effect on fitness that's quantified by this thing called the selection coefficient, or little s here. And the idea is these selection coefficients may have some distribution. And so this is one hypothetical distribution where there's many neutral mutations and many strongly deleterious ones, in other words, ones that reduce fitness quite dramatically, and fewer that are intermediate. Another hypothetical distribution of fitness effects could look like this red curve, where you have many mutations that are almost neutral but maybe slightly deleterious. And there could, of course, also be beneficial mutations as well. So trying to quantify what this distribution of fitness effects looks like is a central goal. A related concept that is going to be much the subject of my talk today is something called dominance coefficients. So imagine here, right, we have genotypes in a diploid organism, and imagine that the ancestral or wild-type state is homozygous. In other words, an individual carries 2 copies of the capital letter, and there's some fitness associated with that genotype, and we're going to call that fitness 1, just essentially arbitrarily scaled to be the most fit genotype. Now, individuals can either be heterozygous or homozygous for the deleterious mutation, the lowercase letter. And so if we look then at the fitness of the homozygote for the lowercase letter, we're going to say that's 1 minus s, where s is, again, the selection coefficient that I was just talking about on the previous slide. So then the question is, what about the heterozygote? And so that's where we get into the concept of dominance here. And in this example, where the fitness of the heterozygous genotype falls exactly between that of the 2 homozygous ones, this would be an additive mutation. And this H coefficient that we'll talk a lot more about, this is the dominance coefficient. And here it's equal to 0.5, essentially saying that the fitness of this heterozygote is halfway between that of the 2 homozygotes. We could have deleterious mutations that are recessive, and that's an example shown here, where now an individual who's heterozygous for the deleterious mutation doesn't have too big of a reduction in fitness. In other words, you need 2 copies of this deleterious allele to really have this big decrease in fitness that's shown here. Of course, mutations could also be dominant, where 1 copy of little a is just as bad as 2, and here h would be bigger than 0.5. So these are some of the key concepts that we'll be talking about, but first I just want to motivate this to say why might we study deleterious mutations, and there's sort of an answer here for everyone. If you're interested in evolutionary types of questions ranging from just general structure of genomes, why they are the way they are, to studying adaptive evolution or conditions under which recombination might evolve, deleterious mutations are essentially pervasive and important for all of these questions. And then if you don't really care about evolution, which this I think would be really bad, but if you don't care about evolution and you're interested in medical genetics, it turns out that, right, the patterns of genetic variation that we see, it's the consequence of the evolutionary process. And, you know, it's currently still, I think, being discussed and debated whether rare or common variants are more important for certain traits and accounting for heritability. And a lot of the models that can underlie or be used to study a lot of the empirical data that's being generated essentially, either directly or indirectly, make some assumption about whether mutations are deleterious or not. And so quantifying that can be a useful starting point for understanding genetic architecture and ultimately designing and interpreting GWAS and other related types of studies. So what are some ways we can study deleterious mutations? So one approach that I'm not going to talk too much about, but I'm just going to mention in passing, is to use these sort of laboratory studies of essentially model organisms. Typically yeast or bacteria can be used, or Drosophila, where you basically start with some ancestral line, keep these multiple replicate populations, allow them to evolve and accumulate mutations, and then essentially measure the fitness of these lines at the end. And typically when people do this, they find, here's just an example from a review paper, that many of the evolved lines, at least in stable environments, tend to have a reduced fitness compared to that ancestral starting material, suggesting that many mutations are deleterious. There's also been models that have been developed to try to estimate some of these selection and dominance parameters from this kind of data. And so, for example, here's one study that looked to estimate dominance coefficients from yeast data. And what they found was essentially this interesting relationship here, where we have the dominance coefficient on the y-axis and the strength of selection on the x-axis. And you can see that as mutations tend to become more deleterious, meaning you move this way, they also tend to become more recessive. I should also say, please feel free to ask questions at any point. And so other studies, experimental evolution type studies, have sort of suggested similar conclusions. But there's a number of problems with these studies. So first is, if you notice on the axes here, or you can still see it, the selection coefficients become quite deleterious. So these types of experimental evolution studies really can only quantify really strong effect mutations. Secondly, they're focused on, in this case, yeast, but also in Drosophila, they can't really be applied to sort of more, any organism. And lastly, and perhaps most importantly, you're limited right to a laboratory environment. So you're not looking at organisms in their natural state. So I think that even though dominance is really crucial, we don't have a very good understanding of the dominance coefficients for most deleterious mutations from these sorts of laboratory mutation accumulation lines. So what's one possible solution? And that is to use, which I'm a population geneticist, and we study natural variation in natural populations. So it shouldn't be a surprise that I would say, well, we should look at genetic variation in natural populations and build models to try to estimate these parameters from the models. So the basic idea is we can look at essentially frequency distribution. I'll go into more specific detail in the next few minutes, but basically look at sites where we want to estimate the distribution and fitness effects and dominance coefficients, and then essentially fit models that include these evolutionary processes to the data. And essentially then find the parameters that maximize the likelihood. So in my talk today, what I'm going to essentially tell you about are first some computational approaches to infer the distribution of fitness effects, then how we can extend those approaches to infer dominance coefficients at the same time. And then the last 2 points move essentially into a bit of the results about application of this approach to data. And then lastly, I'll tell you a bit about, if time permitting, essentially our new model for understanding why deleterious mutations may, not to give away too much of the punchline, but why deleterious mutations may tend to be recessive. So let's start with talking about how can we infer a distribution of fitness effects from data from natural population and some of the tools that others and us have developed to look at this. So the main thing that I'm going to focus on in a lot of my talk is something called the SFS, or site frequency spectrum. And it's a summary statistic of genetic variation data. So imagine here, this is DNA sequence data. This is essentially the data matrix that you'd have where different individuals are the rows and columns represent the variable sites in the sequence alignment. And so you notice these positions here, these are just arbitrary locations in the sequence. The sites where everyone's the same are not shown here. Okay, so the other thing to note is there's these columns, there's two colors, right? The yellow denotes the ancestral sequence, and the red denotes the mutated base. So at this site, A was the ancestral allele, and then it mutated to a G at some point in the history of the sample. So what we can simply do is just tabulate what's the frequency of the mutant allele here. So in this case, it's 1 out of 5. Here it's 2 out of 5. The third snippet would be 1 out of 5, and so on and so forth. So each variable site has a frequency associated with it. That's what's shown on the bottom there. And so the site frequency spectrum is just a histogram of these mutation frequencies. So for example, there are 5 sites where the derived allele is at frequency 1 out of 5. Those 5 positions are denoted by these blue boxes here. Then you can see that there are 2 sites where the derived allele is at frequency 2 out of 5, 2 sites where the derived allele is at frequency 3 out of 5, and no sites where the derived allele is at frequency 4 out of 5. So basically, we can take the sequence data and summarize it by this site frequency spectrum. And we can do this from any empirical data set. We can do it for any class of mutations that we're interested in. And obviously, we're then going to generate this from a model to compare to the data. But we'll get to that in a minute. So why is this a sensible thing to do if you're trying to study natural selection? Well, it turns out this site frequency spectrum is very sensitive to the effects of selection. So what we're showing here are a bunch of site frequency spectra. Here is the predicted frequency spectrum for a neutral, no natural selection, constant population size, essentially just the simplest model you can have. Then as we move from left to right on this slide, mutations are, so this is from a model here, and I'll talk a little bit about that in a second, but the basic idea is as you move from left to right, mutations become more and more deleterious. And so you notice sort of 2 things happen, right? One is you see there's just far fewer genetic variants over here, fewer polymorphisms. In other words, the heights of the bars are all much lower. And number 2, of the genetic variants that are still here, there's many more of them tend to be at low frequency. And so intuitively, that should make sense, right? If mutations are deleterious, natural selection's acting to remove them from the population. So you see there are fewer of them, number 1. And those that haven't been quite removed yet, they tend to be rarer. So that's what's going on here. And so the idea is this site frequency spectrum, it's determined by essentially the strength of selection. Now, there's some interesting subtlety here, and that is that natural selection can affect this frequency spectrum, but other evolutionary processes, like population history, like did the population grow or did it shrink or change size or that sort of thing, those can also impact this statistic. So we have to be a bit careful and thoughtful about how we deal with that. And the kind of approach that researchers have typically taken and that we've also used is to say, okay, let's take one category of sites that we think are actually neutral. So we think they're not under selection. Let's use those to estimate this demographic model. And then let's look at the category of sites where we want to infer selection, but keep that use that demographic model that we estimated previously. In practice, we're focusing on coding regions of genes, right? So and if you throw mutations into coding regions, some of them don't change the amino acid, and some of them do. Those mutations that do not change the encoded amino acid are what we're calling synonymous mutations. And those mutations that do change the amino acid we're calling non-synonymous. So the idea is the non-synonymous category is affected by demography and natural selection. And this is the category we want to infer the parameters of selection and dominance coefficients. Whereas the synonymous sites, we're assuming, and it's questionable whether, to how good this assumption is, but at least to a first approximation in many organisms, synonymous sites are thought to be more neutrally evolving. So we'll use them to infer the demographic history, and then conditional on that, you estimate selection for the non-synonymous ones. Question?
Quickly,
yeah.
What is the scale of resolution are you doing? Is it whole organism, 1 gene, 7 genes?
Great question. Yeah, so this could be, in principle, it could be any of those. Probably more than 1 gene because you need some genetic variation to be there. So I'll be talking about applying this to the whole genome, but then we actually do divide things up into categories of genes to try to get a finer resolution and learn about some biology. So I would say at smallest scale, probably tens of genes would be the finest you could go. OK, so right, then we want to infer the parameters. And I'll talk in a little bit more detail about how that actually works. So what do we need here? So we need a couple things. One is we need a way to generate this expected site frequency spectrum for a population genetic model, right, that includes selection, dominance, and demography. And I'm going to call this x, well, here it's a scalar, but that's ultimately is a vector. That's the entries in the site frequency spectrum. So x sub I would be, for example, if I was 1, that would refer to the number of SNPs that are at frequency 1 in the sample. And then we need a way to assess the fit of the SFS from the model to the empirical one. So the approach that I'm going to tell you about that we're using, was originally developed by Sawyer and Hartle in the early 1990s, called the Poisson random field model. And the basic idea is that the assumption is that mutations enter the population each generation, essentially according to a Poisson process. And they have a specific rate at which they enter. And then each mutation essentially follows its own independent trajectory. So that's essentially what's shown here by these colored plots. And the idea is we can use a simple Wright-Fisher model of evolution for each mutation independently. So what's a Wright-Fisher model of evolution? Basically, this is a model that allows, well, in the strictest sense, the original sense, it basically allows for mutations to change in frequency randomly due to the fact that not all individuals are reproducing. And essentially, you have binomial sampling each generation, allowing allele frequencies to change. We're adding in, essentially, we have that random component, the genetic drift, but adding on top of that natural selection, which is a deterministic force. And that's what's going on here, for example. So how does this actually work in practice? So here's essentially the main, and these are some of the key references that came before we jumped in that derive some of the key stuff here. So the idea is this is coming from our model of evolution. We can then say, OK, how many SNPs at frequency I would we expect to see in our sample? Right? That's what we're going to find here. And we're writing that as a function given the demographic model. That's what I'm having this capital theta d, and the distribution of fitness effects. Yeah? Yeah. Yes. Yeah, so yeah, good point here. So if you look, right, you see for the deleterious mutations, they all tend to be at lower frequency, and that's because of purifying selection keeping them a bit rarer. Yeah, great question. OK, so what are we actually doing? So what we have here is this part here just represents the probability of a particular selection coefficient given our distribution of fitness effects. So these are just basically conditional probabilities here. Then in the light blue, we have the probability of a certain frequency, of an allele having a certain frequency in the population, given the demographic model and the selection coefficient. And then lastly, this should look a little bit familiar as essentially a binomial distribution. And so what that's doing is that's going from our population frequency x to our actual integer count in the finite sample of size n chromosomes in our actual data set. And then we integrate over the frequencies at x and the distribution of fitness effects. So where do these parts come from? I mean, this essentially comes from just numerically integrating whatever distribution we assume for fitness effects. This part here essentially comes from a diffusion approximation to this Wright-Fisher model. We don't have time to get into the details here, but there's existing software packages that essentially implement that part, and we leverage that here. Oh, and lastly, this is just, yeah, the theta over 2 is the expected number of mutations entering the population. OK, so what do we do then? Well, basically, the idea is each entry in the frequency spectrum is going to be essentially Poisson distributed with rate parameter equal to the expected value shown, or the expected value found from the model, essentially on the previous slide. And then that allows us to use a Poisson likelihood function, right, from the parameters of the distribution of fitness effects. So the idea here is when we want to use this approach for inference is we essentially optimize this likelihood function and find the parameters of this distribution of fitness effects that maximize the probability of the observed frequency spectrum, or the X vector, if you will. Importantly, this is using information both about the relative proportions of the SNPs in the sample, as well as the overall absolute number of SNPs. So we do need a good idea of what the mutation rate is for this to work. And that is a challenge of this particular approach, but you do gain a lot of information because very strongly deleterious mutations will never be segregating in your not hugely sized sample from the population. And so by not seeing them, then that tells you something about the more strongly deleterious mutations. So how do we actually apply this? So the idea is we use essentially the synonymous sites to estimate this demographic model here, the theta d. And then we keep those parameters fixed. And then we take the frequency spectrum for the non-synonymous sites and then infer the parameters of the distribution of fitness effects here. And we use or can consider a variety of different functional forms of the distribution of fitness effects. The gamma and log normal are some of the more popular ones in the literature and seem to work quite well, although some mixture distributions do fit a bit better. That's sort of a separate talk, but the basic idea is we can fit some sort of parametric or even, I guess, more of a non-parametric distribution to the data. So we've applied this approach essentially to study the distribution of fitness effects, and I'm not going to, I don't have time to get into the specific details here, but in some work we did a couple years ago, we used this type of framework to actually test, do these distributions of fitness effects differ between species, starting with comparing, and what's shown on the slide here, humans and Drosophila, again focusing on amino acid changing mutations. And the punchline maybe comes from this figure here, where I'm showing the proportions of new mutations that are essentially not so deleterious over here and very deleterious over here. And what you can see is flies are in red, humans in blue. You can see these distributions look quite different. And statistically, they're quite different as well. And so it's suggesting, in fact, that different distantly related organisms do have different distributions of fitness effects. And there's a lot of reasons why we think that's the case. And I'll point you to that paper, or I'm happy to chat more about that later. But what I want to focus most of my talk on today is actually dominance coefficients. And so can we extend this approach to actually infer dominance, as well as just a distribution of fitness effects? I should point out the vanilla theory that I've talked about so far assumes that mutations are all additive. And the result here, this is also assuming mutations are additive. So why not just use the same approach, but add an extra parameter for the dominance coefficient, right? That's sort of the obvious thing to do. There's some existing theory on this. And one of the things is that it's quite challenging. So what I'm showing you here are 3 different site frequency spectra for different dominance coefficients, but changing what the selection coefficient, little s, is for each of these. And what you can see is the punchline is these frequency spectra all look very similar to each other. So the idea is we have an identifiability problem where h, the dominance coefficient, is confounded with s, the selection coefficient. In other words, different h and s values, their products can be the same, And that's what's essentially determining this frequency spectrum in a single outbred, outcrossing population. So the problem is if we want to be able to infer S and H together, we don't really have much power to do it using the sort of approach that I've been telling you about. So our idea was, well, wait a minute. What if we, instead of using just one population that's essentially randomly mating, what if we also look at, for example, a population that's mostly selfing? In other words, what we mean here is, like particularly think about plants here that are self-fertilizing. Then what would we see? So what we did was we said, OK, let's take the same values of S and H that we used here, simulate some data now in a population that's predominantly selfing. Do the frequency spectra look the same? And you can see here they don't. They actually look very different from each other. So what that tells us is, OK, maybe if we consider a pair of populations where one is outcrossing and one is largely selfing, then we're better able to disentangle selection from dominance. So what's some intuition as to why this works? Well, in outcrossing species, right, if we're thinking about deleterious mutations, they're likely to be rare. And rare mutations, just by virtue of basic Hardy-Weinberg equilibrium theory, would say that most rare mutations will be found as heterozygotes. And so, OK, if most deleterious mutations are found as heterozygotes, that gives us information about the product, HS. Now, if we go to a species that's very highly inbred or highly selfing, by definition, most genetic variation there is going to be homozygous, right? That's essentially what inbreeding or selfing will do, is it leads to an increase in homozygosity. So in this case, we're getting information about just S. H doesn't really matter in the selfing population because you're not having these homozygous, or excuse me, we don't have heterozygous genotypes. And so the idea is if we combine that data together in a way I'll talk about in a second, then that gives us information about H and S essentially independently of each other. So this is work that was done by Christian Huber, former postdoc in the lab, and Arun Durvasula, who's a genetics and genomics graduate student in my lab. And Arun's here somewhere. There's Arun. So if you have any questions, direct them to Arun. I'm kidding. Do feel free to ask Arun questions about this work. OK, so how do we do this then? Well, what we can do is essentially we're going to use the same Poisson random field theory, extend it in two ways, right? First, including the dominance coefficient, but then also including the two population thing I just told you about a few minutes ago. So the idea here is this is again, we can write down our expected frequency spectrum as a function of demography, distribution of fitness effects, and now this theta h, that's referring to our dominance parameters, which I'll make clear exactly what I mean by that in a second. We're going to use essentially the same likelihood framework as before, but now our model has this extra parameter, the theta h. And the trick is that, OK, how do we apply this and extend it to work with two populations? If our populations are sufficiently diverged, we can assume they're independent of each other. And in that case, right, then we can say that if the frequency spectra are independent between the inbred and outbred population, we can essentially, working in log space, sum up the frequency, or sum up the likelihoods of the model given the data together for the combined likelihood of the parameters. So the idea is this would be the log likelihood function, the Poisson log likelihood function for the inbred population, and then this is the one from the outbred population. And then, right, so then, OK, this is again extending what I told you about before, where now we have our dominance coefficient added in here. This is really the only step that adds in the dominance coefficient when we want the population allele frequency given demography, the selection coefficient, and the dominance coefficient. So Christian came up with a clever trick here. So to calculate the probability of the frequency of the allele frequency given the model parameters, I mentioned we leveraged this existing software developed by Ryan Gutenkunst called DADDY, and that numerically solves the forward in time diffusion equation. And that works really well. It's really great. The problem is now when we're considering many pairs of S and H, we essentially are generating this massive lookup table of values of frequency spectra for different combinations of S and H. And when we evaluate many, many points on this grid, it does become computationally quite expensive. And so what Christian decided to do was actually do a coarse grid of 50 by 20 values. So, when I say gamma here, that's the selection, essentially the selection coefficient scaled by the population size. So just think about it as the strength of selection. And so use a coarse grid and then cubic spline interpolation. And so here's some examples of how this works, where on the x-axis we have the strength of selection. Up this top panel, dominance coefficient of 0. And this is just looking at the number of singletons that you would expect to see. The red points denote grid points that we actually perform the evaluations using DADDY. And the blue line denotes the interpolation. And you can see across different, these are just a few arbitrary parameters to show you an idea of how this works. You can see that it works, it actually works quite well and dramatically speeds up how this could actually be implemented. Okay, so now in the last sort of 15 minutes or so, I'll tell you about how all this plays out and what we find about what we can learn about dominance coefficients. So I mentioned, right, we need to apply this approach to selfer and outcrossing population. So plants are a logical choice. And so we focused on looking at Arabidopsis, where we have the sort of common laboratory strain or species, Thaliana, which is predominantly selfing. And there's a related species, Arabidopsis lyrata, that's predominantly outcrossing. And so we can look at whole genome sequence data from these two different species. And then we essentially look at a few different models of dominance, right? So our simplest model is an additive model where all mutations have a dominance coefficient of 0.5. Slightly more complex, all mutations have the same dominance coefficient, but it's something other than 0.5. And lastly, that there's some, the sort of most complicated model, that there's some relationship between H and S. And so this is one functional form that we looked at. We looked at a few others as well. But the basic idea here is that this dominance coefficient now depends on the selection coefficient. And so it could look something like this green line here, where, as previously suggested in experimental studies, more deleterious mutations tend to be more recessive than less deleterious mutations. So the idea is we will try to fit all of these different models. And what do we see? So the beauty of these is they're nested models, so we can use a series of likelihood ratio tests to essentially compare the fit of them. And so first, obviously, we test the additive model versus the constant H model. And we find, in fact, that the constant H fits better. And I believe it's not on the slide, but our point estimate was 0.46. So slightly recessive, but not too much. But then if we compare the additive model to the HS relationship, you see that that actually fits much better. And it fits better than the constant H model. OK, so what is this HS model that we find? This black line essentially denotes that relationship between the selection coefficient and the dominance coefficient. The blue bars is the cumulative distribution of fitness effects. And so what you can see is that as mutations become more deleterious, so as you move this direction, they become more recessive. So a couple things here. Mutations largely are recessive, number one. And number two, as they become more deleterious, they become more recessive. Just to sort of put a number on this, we estimate about 15% or so of mutations are more deleterious than 0.001 and are more recessive than, or have an H value smaller than 0.025. OK, so there are a lot of things that we looked at to say that could be confounders that could bias the inference. I don't have time to get into all of them, but I'll just tell you about a couple that I think are kind of interesting. We fit a variety of other functional forms here beyond the one I showed you on the previous slide. Here's what they kind of look like. So Sure. In this range, they're a little bit different, but the idea of them decreasing, H decreasing as mutations become more deleterious, that tends to be pretty robustly supported. You'll notice there's something going on with the intercept there, which I'll tell you about that actually in a couple minutes. But we also did simulations, right, under the, as one would do with developing a new population genetic method. You do simulations under the model and look at the behavior of the test statistics. So this is looking at the behavior of the likelihood ratio test statistic, where we essentially simulate data under the null hypothesis where things are additive, or the constant H model, so that's not the null. And then we fit the constant H model there and compare that to the additive model, essentially. And so this is then our distribution of the test statistic under the null, follows this green line here. It's close to the asymptotic chi-square distribution, although the tail is a bit heavier. The constant H model, you can see here that the likelihood ratio test statistic is shifted very much to the right, which is what you would expect, indicating we have good power to tell them apart. This is now the same thing, but with the HS relationship model. And so again, here we compare we fit the HS relationship model to both the null and the HS relationship data and look at the likelihood ratio test statistic and see that, again, we get good separation. So, OK, quick thing here. You notice that as mutations become almost effectively neutral, it looks like they become dominant. In other words, the H coefficient from the black line that we infer goes up past 0.5. So does that actually mean that nearly neutral mutations are dominant? At face value, that's what it seems to imply. But biologically, that's probably not very reasonable. So we essentially said, OK, well, we're assuming, right, that for a particular S, there's only one dominance coefficient. In reality, there could be a distribution of dominance coefficients for a given selection coefficient, and we're essentially ignoring that. So could that be creating some sort of bias? So what we did was essentially simulated data where we had a distribution of H for any given S value, and that's essentially these red points are reflecting that. So we assumed H came from a beta distribution, just to pick some distribution that would give us some noise. And then we essentially applied our inference method to the data. So what you can see here in blue is the actual average fit to the simulated data before we do any inference. And then the green curve comes from our inference. And so the good news is it's getting the blue curve quite well, down here. But if you look over, you see that it overshoots this intercept parameter. So what we think is happening, actually, is there's an upward bias in the intercept parameter that's coming from data where the observed maximum here is, or the average here is 0.5, meaning additive. Yes?
We just simulated the mean without noise, but from a different functional model than you are simulating. Wouldn't you generate the same effect? Because if you're just estimating a function, so
yeah.
if it fits very well down there, it might not fit the string.
Yes, I don't know if we didn't try that particular simulation. We did try fitting other functional forms to the data where we constrained it at 0.5. And that didn't fit as well as the one where we let it go higher or let it be a free parameter. But it still fit better than the other, certainly better than the additive model.
But your functional model is a global model. So
right.
There's no requirement that it is equal everywhere.
That's right. All right, so in the last, I'm kind of short on time, so I'll tell you, let me jump ahead to the conclusions. And ask Arun why the mutations are recessive. So most deleterious mutations are partially recessive, these are some of the first estimates of dominance coefficients from molecular population genetic data in natural populations. And the part that I didn't tell you about is that, so we think we can explain these patterns that we see in the dominance relationship based upon a model where essentially genes have different optimal expression levels, and then when they are expressed at their optimal expression levels, then we can get dominance essentially arising as a consequence of that, where mutations that are more highly expressed, or in other words have higher optimal expression levels, will tend to look more, mutations in those genes will tend to be more additive than mutations in genes that have lower optimal expression levels. And that we see these patterns in the data. So any other questions? All right, well, I'd like to thank Christian and Arun for doing a lot of the inference work, and a key collaborator, Angela Hancock, for helping us with obtaining some of the data and funding sources. And I'll be around and happy to chat more offline. Thank you.
One or two questions on this one, Peter? A couple of minutes for a copy.
Yes. Sure. I can give the sort of 2-minute explanation. Basic idea is something like this. So empirically in the data, we see a pattern like this, where genes that encode structural proteins tend to look slightly more additive than genes that make up enzymes. And we see more broadly that genes that just have higher expression levels tend to look more additive. So the idea is we classify genes based on the expression profile and then redo the inference on the different categories. That's essentially what we find. And so the idea is, okay, what can explain that sort of pattern? And turns out in the literature there was a model basically that says fitness f of x as a function of gene expression level, which here is denoted x. So we extended this model in a couple ways. Basic idea is here's the fitness function as a function of gene expression. The star is the optimal expression. The idea is then a heterozygous mutation decreases expression by a half in this model. And so if you shift essentially over a half, it's not really a big change, just based on the shape of this curve. So this would be a recessive mutation because you need 2 copies to really take the hit in fitness. Whereas a gene that has higher optimal expression, so that's denoted by the orange star here being further out, if the curve fitness function looks something like this, then a mutation that decreases expression by half will have a bigger impact on fitness. I mean, it's not entirely additive, but it's more additive than what's seen over here. And so the idea is, there's also a cost to gene expression here. So the idea is a fitness cost. So it's not just that it runs away, and you'd keep increasing fitness with increasing expression. So the idea is if we simulate from this model, that's what's shown over here, we recapitulate this relationship between H and S, number 1, and number 2, genes that have higher optimal expression level will tend to look more additive. And that's similar to what we see in the empirical data. So that's the quick version of what we think could explain the patterns of dominance in different gene categories. Yes?
I wonder whether you can make a messing point if that needs some work done.
Yeah, I think there has been, not so much with these sorts of molecular population genetic approaches, but yeah, that definitely could be something fun to think about. Yeah. All right, thanks.